other part of this lecture, which is the somatic sensation. And the first thing on the somatic sensation, we're going to discuss the tactile and position sensation. These are part of our general sensation. Our objectives is to list the type of mechanoreceptive sensation, mechanical sensations, and its receptors. Describe the two pathways for its transmission. One pathway is called dorsal column medial lemniscal pathway tract, and the other is anterior lateral spinotheramic. Follow these pathways to the cortex and differentiate these two pathways. What is the differences between them, because they are different pathways, different systems. Okay, classification of the somatic sensation. The somatic sensation can be classified into mechanical according to the type. Then here according to the type. Mechanical or mechanoreceptive sensation like tactile, touch, pressure, vibration, tickle, and itching. And position or proprioceptive sensation, static position or rate of change, dynamic here, static or dynamic position. The other thermoreceptive sensation, temperature. They detect change in temperature. Usually these receptors, they detect changes in the stimuli, in the stimulus. They don't detect the they don't detect the stimulus. They detect any change. Like detect heat and cold, warm and cold. Nociceptive, these are for pain receptors. Detect pain and activated by any factor that damage the tissue. Pain is usually due to the damage of tissue. Tactile receptors. The tactile receptors are usually they are free nerve endings, and the free nerve endings are connected to the A delta fibers or C fiber. A delta, this is the smallest myelinated fibers. You know A is myelinated. Myelinated fiber are A fiber. C is unmyelinated. These free nerve endings, they detect touch and pressure. They are found everywhere in the skin and other tissues. Meissner corpuscles, they are connected to the A beta fiber. This is large myelinated fiber. They are rapidly adapting, since Meissner's usually encapsulated receptors, they are rapidly adapted within fraction of a second, and detect movement of light object, light in something that's not heavy, light object in the skin, tactile, found on non-hairy skin or called glabrous skin, and they are found in the fingertips and lips. Merkel's discs, they are connected by A beta fiber, so they are also rapidly respond rapidly at first and then slowly adapting. Detect steady state, and they are found on hairy and non-hairy skin. So they detect touch also. Hair end organs, they are found around the hair shaft. They detect the movement of hairs. Raffini end organs, they are found deep in the dermis. They detect changes in pressure and joint formation. Slowly adapting Pacinian corpuscles, they are found down in the dermis, and they are rapidly adapting because they are corpuscular, and they detect vibration and pressure. They are connected to A beta fibers too. These are the receptors. These are the free nerve index down in the epidermis. This is extended TEBS receptor, tactile hair receptor. Pacinian corpuscle down in the dermis. They are like onion rings here. And this is the Messner corpuscles down in the epidermis. Krause's corpuscle down in the dermis. Raffini end organ around the joint and in the dermis. Golgi tendon apparatus or Golgi tendon organ and muscle spondyle, these are found in the skeletal muscle and on the tendon. We were going to discuss them when we discuss the motor system and the spinal cord. This is the ego-dome receptor, which is a division from the Merkel's disc. Merkel end organ. Then they divide and form a dome under the epidermis. Dome, that's called ego-dome receptor. Ego-dome receptor, the multiple number of Merkel's disc, myelinated and operating under the epithelium, and they detect the touch. Now the Messner corpuscles are hair receptor, Pacinian organ, Raffini and organ. They transmit signals in type A beta, that's very fast, between 30 and 70 meters per second. Free nerve endings transmit signals through A delta and C between 5 and 30 meters per second, some by C, which is unmyelinated, between 0.5 to 2 meters per second. The more critical information, the more the critical information is, the faster the rate of transmission. So fine touch, fine pressure, they are transmitted very fast through A beta or A alpha sometimes. Now the pathways for transmission of sensory information. Almost all sensory information enter the spinal cord through the dorsal roots of the spinal cord. So the afferent neurons, they have their cell bodies at the dorsal root ganglia, and they enter the spinal cord, and then when they enter the spinal cord, they ascend in the spinal cord as two kinds of tracts: dorsal column, medial lemniscal tract. These tracts, they ascend in the dorsal column of the spinal cord, and they form the medial lemniscus. That's why they are called dorsal column. You know the spinal cord like here. So this is a posterior aspect. And this is the anterior. Now the spinal cord has two kinds of matter. They have the gray matter here, gray, and white matter outside. The white matter by the H-shaped gray matter having two kinds, having the columns. So here we have the columns. Here we have the posterior column or dorsal column, and we have here the lateral column, and these columns consist of collection of axon, lateral, and here we have the anterior column. Anterior or ventral column, it's the same, dorsal or posterior, so we have the anterior column. So the dorsal column, the medial lemniscus system is located here in the dorsal column. They form the medial lemniscus. The other system is the anterior lateral column, which is the anterior lateral system, which is going to be formed here in the anterior and lateral aspect of the spinal cord here. This is the dorsal column system. Okay. Now, first of all, the dorsal column medial lemniscus system. The dorsal column contains large myelinated nerve fibers for fast transmission. They're usually A beta fibers. Since they have very large fibers, they have very high rate of transmission. They have very high degree of spatial orientation. So the fibers, they come from each part of the upper body is arranged in a very organized way. So this is called high degree of spatial organization. Spatial, it means position, and throughout the tract, this is called spatial fidelity, and they transmit information rapidly and with a high degree of spatial and temporal time. So spatial, position, place, and temporal time fidelity, faithfulness, i.e. discrete types of mechanoreceptive information. What they transmit fine touch, fine pressure, vibration, position, and stereognosis. We will come to that in a minute. Okay, so this is the characteristic of this type of tract. This is the pathway of this tract from the receptors. The afferent neuron enters. Its cell bodies are found in the dorsal root ganglia. Then it enters the posterior horn. The posterior horn is usually sensory horn. And then they ascend until they go to their respective nuclei. You know, this tract here consists of two parts. One is called fasciculus gracilis, الحزمة النحيلية, and fasciculus cuneatus الحزمة الوتدية. So there are two fasciculi. The fasciculus gracilis are the medial one, and the fasciculus cuneatus are the lateral one. You look here to the lateral column here, say. In the lateral column we have the gracilis here and the cuneatus here. So here fasciculus gracilis and here fasciculus cuneatus. So here FG fasciculus gracilis and here FC fasciculus cuneatus. So the fasciculus gracilis transmits the information from the lower part of the upper body. The fasciculus cuneatus transmits the information from the upper part, from the lower limb fasciculus gracilis, from the upper limb fasciculus cuneatus. They run until they reach their respective nuclei in the medulla. In the medulla we have cuneatus nucleus and gracilis nucleus. Each of these tracts, they synapse at the gracilis nucleus, and the fasciculus gracilis synapses at the gracilis nucleus. And the second order neuron, it crosses the midline in the medulla oblongata and ascends as the medial lemniscus, and they go to the thalamus, yes. And in the thalamus they synapse at the ventrobasal complex. The ventrobasal complex consists of two parts, VBL, ventroposteriolateral, VBL, and VBM, ventroposteromedial. This is called ventrobasal complex. And from the ventrobasal complex they go, and third order neuron, they go where? They go to the somatosensory cortex. Primary somatosensory cortex. Somatosensory cortex. So this is the pathway, this is the tract. This tract called the dorsal column medial lemniscal pathway. So this is the posterior column or dorsal column medial lemniscal pathway, convey information of nerve impulses for fine touch, for fine pressure. What do we mean by fine? Fine, we mean well localized. Exact localization is very important. And why exact localization here? Because it transmits its information through rapidly transmitting neurons. Fine pressure, fine touch, vibration, weight discrimination. To discriminate, if you put something at your hands, you discriminate this is heavier or lighter than the other one. Two bones discrimination, to compare, to see if the stimulus is stimulating 2 points or 1 point. This is what we call it 2 points discrimination or fine discrimination. Stereognosis to discriminate by touch, by moving your body or your hands over an object, you can determine its smoothness, its form, and its shape. So you use the touch information, the fine touch information, to determine the form and shape of the objects you are moving your body over it. And this is the essence of the Braille language for the blind. So what the blind they do, they move their fingers, and the fingers, they have very high density of receptors. They move their fingers over knobs, and these knobs, according to the number of knobs, they can determine the from their experience, they can determine the letters, and accordingly they can read the letters and read the language. Conscious proprioception, which is the sensation of position from the limb, trunk, neck, and posterior head to the post-central gyrus of the cortex. So these are the receptors for touch, pressure, vibration. So the receptors for fine touch, fine pressure, vibration, stereognosis from the lower limbs of trunks. They enter the spinal cord in the posterior horn. They ascend in the dorsal column, forming the medial lemniscus for the gracilis, fasciculus gracilis, and fasciculus cuneatus. They go to the medulla oblongata. They synapse at their corresponding nuclei. The fasciculus gracilis synapse at the gracilis nucleus, and fasciculus cuneatus synapses at the cuneatus nucleus. Then they cross the other line. They decussate. They cross to the other side. And they ascend, forming the medial lemniscus, where they go to the ventrobasal complex of the thalamus, ventral posterior nucleus, VBL part of the thalamus, and from there they go to the primary part of the cerebral cortex, which is in the post-central gyrus. Post-central gyrus, we will talk about where the post-central gyrus in a minute. Okay? Remember here, fine touch or fine pressure, well localized. Okay? Remember, stereognosis, it's to discriminate things by moving your hands especially over it, and you determine its smoothness or roughness or the shape. Is it there is angle or there is roundness, whatever. And this is the way that the blind use this kind of sensation, and in the blind it is highly developed sensation because they use it too much. And these are transmitted at the dorsal column. So receptors here for the dorsal column. We will talk about the other system in a while. The other system is the receptors here the same. This is the receptors for gracilis, the same for cuneatus from the upper part of the body. Receptor for touch, pressure, vibration, proprioception, and stereognosis too. Stereognosis, and from the upper limb, they are transmitted through the fasciculus cuneatus. It's drawn on the other. Remember that these tracks are found in both sides, but what it is drawn here, they are drawn in one side here, gracilis, and the other side is cuneatus, just to illustrate it only. But the gracilis and cuneatus found here, gracilis and cuneatus found in each side. And the crossing, it occurs for both tracks, gracilis or cuneatus, at the level of the medulla oblongata. So remember here, there is crossing. So the information or the sensation that are coming from the right side here, say right, is going to where? Is going to the left side of the cerebral cortex, and vice versa, information coming from the left side of the body going to the right side of the cerebral cortex. Okay? That's why it's a brain here. When you say about the brain, the left brain and the right brain, they cannot work by themselves. They have to work together, because the information from our right side of the body, they are laid where? They are laid to the left cortex. So you cannot split the cortex. You say this left cortex, it's controlling the left side, or the other way around is the right cortex. It controls, it receives information from the left side of the body, and the left cortex receives information from the right side of the body. Due to the crossing that occurs here in this system, it occurs at the level of the medulla oblongata. This is the cerebral cortex. The cerebral cortex, this is a lateral view of the cerebral cortex. Lateral view of the cerebral. This cerebral cortex is divided into lobes by sulci, sulcus. It's plural of sulcus. This is the central sulcus, and this is the lateral sulcus or lateral fissure here. It divides the cerebral cortex into 4 lobes: frontal lobe, anterior to the central sulcus here, and we have parietal lobe posterior to the cerebral sulcus, and we have the parieto-occipital notch here that also separates the parietal lobe from the occipital lobe, and down inferior to the lateral sulcus or lateral fissure we have the temporal lobe. We have 4 lobes. Now here, posterior to the central lobe here, this area posterior to the central lobe, we have what we call it post-central gyrus. Post-central gyrus. This post-central gyrus is the primary somatosensory area. So all our somatosensory sensation, they come to the post-central gyrus. What is a gyrus? So here, when you talk about sulcus, this is sulcus here, and there's another sulcus here. The area in between, this is sulcus, and this is sulcus 2. The area between 2 sulci here is called gyrus. So a gyrus is an area that's between 2 sulci. So here is the area of the post-central gyrus, where the somatosensory sensation are laid down, and there is very high organization, very important representation in this post-central gyrus area. This is the somatosensory cortex that was called area 1. Remember here there is very high represent very high level of organization. And there is another area posterior to the area 1 is called somatosensory area 2, which has very gross representation of our body. Look to the presentation of our body here at the cerebral cortex. This is the representation. This is frontal section of the primary somatosensory area. Look to the representation. This is the lateral aspect. The representation is upside down. The face is down and the legs are up. So it's upside down. And the representation is contralateral. The representation on the right side is the representation of the left side of the body. And the representation on the right side is the representation of the left side of the body. The other things, the area in the cerebral cortex that represent any part of our body is not proportional to the size of that area on our body. It is proportional to the number of receptors on that area. Number of receptors. So look to the hand here. The hand, the representation, the area that represents the hand in the cerebral cortex is much larger than the area that represents the lower limbs. Why? Because the number of receptors in the hand is much more than the number of receptors. Let's to the lips here. How much are represented? the tongue, because so the size of the cortical region that representing any part of our body depends on the density of the receptor on that part, density of the receptor. And so it is contralateral, it is upside down, and the area in the cortex is proportional to the density of receptors in the area of our body. Look here, the same thing here, our face, lips, hands, upper limbs, fingertips, lower limbs, very small area. So this is the representation. This is look to the area, arm area, how big the area of the arm, how big the area of face, how small the area of trunk and lower limb, because the number of receptors in the arm area, the number of receptors in the face are much than the number of receptors on the other areas. Now look to the dermatomes. Where is these dermatomes? This is very important when we talk about pain also. This is anterior aspect of the body and posterior. You remember from your embryology background that the embryo started as a tube, and this tube consists of segments. And this segments consist of 3 layers: endoderm, mesoderm, and ectoderm. And from these, they are the spinal cord segments are developed, and we have 8 cervical segments. We have 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. You remember from each segment there are 3 layers. The nerves, they are of the nervous system, it's developed from the ectoderm, and the mesoderm get the soft tissue, the connective tissue, and the endoderm give the epithelium. So if you talk about here the C8 T1 T2 here, C8 T1 T2, this is where the heart develops as soft tissue, as connective tissue, soft mesoderm, and this is the what supplies the hands here, the hands. So the heart comes from, say, C8 T1 T2, and the hands, the medial aspect of the hand is supplied by C8 T1 T2 nerves, that the same segment from where the heart. So these are the dermatomes, and as you see from these dermatomes, if you have, say, a loss of sensation at this area here, this is the area of L5, then you know that there is loss of or abnormality at the between L5 and L4. Say sometimes the nerves that supplies this comes from L5 or L2, or and this is important for when we talk about pain. So T10, where the umbilicus develops, and this is where the appendix is developed. We're going to talk about it when we talk about pain 2. The anterior lateral system pathway. The anterior lateral or anterior spinotheramic pathways comes and they run in the anterior and lateral aspect of the spinal cord. We might have anterior spinotheramic and lateral spinotheramic. They run through a small myelinated and unmyelinated A delta and C fibers. They have low degree of spatial and temporal fidelity orientation. Why? Because they are slow conducting fibers. So the degree of fidelity or the degree of orientation is very low, and there is a lot of divergence. That's why that the system is not as accurate as the, so that's why they transmit crude sensation, crude touch, crude pressure. What do we mean by crude here? We mean poorly localized sensation. On the other hand, they transmit broad spectrum of modalities. You remember the first system, the dorsal column system, transmits one kind of modality, which is mechanoreceptive sensation, touch, pressure, proprioception. كلياتهم they depend on mechanical sensation, mechanical receptors. Here this system transmits, beside this mechanical sensation, crude touch, crude pressure, tickle, itching. These are tactile sensation, mechanoreceptive sensation. They transmit pain and temperature sensation, and these are very important sensations, especially pain. Pain is very important sensation for our body because pain is protective in nature, and pain is due to damage of tissue. If you feel pain, it means you can stop the damage that occurs from the tissue by removing your organ from that damaging stimuli. Now this is the pathway for this system. From the dorsal root ganglia, from the receptors, they enter the spinal cord at the posterior horn, and in the posterior horn they synapse. Usually they go 1 or 2 segments up or 1 or 2 segments down. Then they cross to the other side, and they cross anterior to the central canal of the spinal cord. And they ascend where? They ascend either in the anterior or division of the anterior column of the spinal cord, or they ascend in the lateral column of the spinal cord. So they ascend in the anterior column of the spinal cord, or they ascend in the lateral column spinal cord. That's why it's called anterior lateral spinotheramic. And where they go to the ventrobasal part of the thalamus, which is VBL, VBM, ventroposterior lateral and ventroposterior anterior. And also, especially for pain, they go to the intralaminar nuclei of the thalamus. Intralaminar. So, besides the ventrobasal, VBL, VBM, they go to the intralaminar nuclei of the thalamus, and from there they go to the postcentral gyrus, which is the primary somatosensory area. These are the tactile sensation. When we talk about pain, we will talk more in details about its pathway regarding pain. Okay. This is the system, the anterior lateral spinotheramic pathway. It conveys impulses for crude touch. Crude, it means poorly localized. For what? For crude touch, crude pressure, pain, cold and warm temperature, itching and tickle sensation. Itching and tickle, especially tickle sensation is important for sexual sensation. From the limbs, from the trunk, neck and posterior head of the postcentral gyrus to the postcentral gyrus of the cerebral cortex. So they go, this is the first order neuron, the receptors for pain and temperature, crude touch, crude pressure, tickle and itching. They enter the spinal cord at the posterior horn. In the posterior horn they synapse. Sometimes they go one segment up or one segment down, and then they synapse where they synapse in the posterior horn. After they synapse, they cross to the other side and they ascend in the lateral and anterior column, where to the thalamus. So the first order neuron synapse at the level of the spinal cord, the second order neuron synapse at the level of the thalamus, and the third order neuron goes to the primary somatosensory area in the postcentral gyrus. So this system is different than the other system. It has broad spectrum sensation. The other system is just mechanoreceptive sensation. This is no pain and temperature. This is different modality. Pain is nociceptive modality, and temperature is thermoreceptive modality. So modality, what we mean with modality is the type of sensation. Type of sensation. Comparison between the two systems. The dorsal column is well localized compared to the anterior lateral, which is poorly localized. The dorsal system has better temporal, time, and spatial place faithfulness or organization or orientation. Because why? Because it transmits at very high speed, 130 to 120. On the other hand, the anterior lateral has very low level of temporal and spatial orientation. The dorsal column has a better intensity grades. So you just can say if you have grades from 0 to 100, the just simulation. The dorsal system uses the absolute grades. So between 0, 1, 2, 3, 4, 500. The anterior lateral system use the lateral grades, use A, B, C, D. So the lateral grades has much lower grades than the absolute grades. So more than 100 grades compared to less than 20 for the anterior lateral. Repetitive stimuli are only confined to the dorsal column. Repetitive stimuli, because why? Because they transmit at higher speed. If it's higher speed, you can have rapidly adapting sensation, but in the anterior lateral they are transmitting too fast, too slow. They cannot discriminate different intensities of small difference between them. Anterior lateral tracts transmit broad spectrum modalities. Here في nociceptive pain, في temperature, besides the mechanoreceptive touch and pressure and so on. Okay. Now from the face there is another system called trigeminothalamic system. Trigeminal, which is the fifth cranial nerve. This is the fifth cranial nerve, trigeminothalamic pathway. And the trigeminothalamic pathway transmits all the sensation from both systems, from the dorsal column and from the anterior lateral column. It then conveys nerve impulses for most somatic sensation from face and nasal cavity, oral cavity, and teeth to the cerebral cortex. It is synonymous to both sensory system, whether it is dorsal, so it transmits fine touch, fine pressure, crude touch, crude pressure, pain and temperature, whatever, from the face, nasal cavity, oral cavity, teeth. So for receptors for touch, pressure, vibration, pain, cold, temperature, and tickle in the face, nasal cavity, then they go to the nuclei of the trigeminal trigeminal nerves. They cross to the other side and they go to the thalamus, trigeminothalamic, ventroposterior lateral, and then they go to the primary somatosensory cortex, which is in the post-central gyrus. Okay, so it is the same. It is crossed tracts. It's not. It's both the same like the other. So it goes like the other tracts. It goes through the. It transmits information for fine touch, fine pressure, and vibration, stereognosis from the face and the nasal cavity, oral cavity, teeth. So this is the trigeminothalamic tract. This is a comparison between these two tracts. This is the dorsal column, medial and muscular tract, fine touch, fine pressure, proprioception, vibration, and stereognosis. In the fasciculus gracilis or fasciculus cuneatus, goes to the medulla, crosses to the other side, ascends to the thalamus, ventrobasal complex of the thalamus, then to the posterior column, to the posterior post-central gyrus of the primary somatosensory cortex. And this is for cuneatus. Fasciculus cuneatus transmits the information from the upper part, from the upper half of the body, and the gracilis transmits the information from the lower half of the body. On the other hand, the anterior lateral spinotheramic pathway, the receptors for pain, temperature, crude touch, crude pressure, enter the spinal cord, and they synapse in the spinal cord, crosses to the other side, they ascend in the anterior and lateral aspect of the spinal cord, go to the thalamus, ventrobasal part of the thalamus, and intralaminar nuclei, then they go to the primary somatosensory cortex. Look here, it is very important to know where is the crossing. The crossing for the dorsal column is at the level of the medulla. So if the transection is found below the area of the medulla, the loss of sensation is ipsilateral, same side of the transection, same side of the damage. Now on the other hand, here, if this system is, the damage is at the level of, if it is half the level of the spinal cord, the loss is on the other side, on the contralateral. But if the damage is above the level of the, medulla, the loss of sensation in both system is contralateral on the other side of the transection, on the other side of the damage. So this differentiates these two systems. The first system, dorsal column, depends if the transection, if the damage is lower than the medulla, it is ipsilateral, same side. If it is above the medulla, it is contralateral, other side. The anterior lateral system, no. If the level of the transection, the level of disease is on the spinal cord, it is contralateral. And if it is in the medulla, if it is above the spinal cord, it is contralateral. Below the medulla, above the medulla, it is both systems, they share the same damage. Somatic sensory cortex, located post-central gyrus, highly organized, distinct spatial orientation. Each side of the cortex receives information from the opposite side, contralateral aspect. Unequal representation, you know. Lips have greatest area representing, followed by face, thumb, because they have the highest density of receptors. Number of receptors or high, number of receptors. Too large number of receptors. And trunk and lower body have very least area because they have lower number of receptors. There are 6 separate layers of neurons in the cerebral cortex, from outside to inside. Layer 1 outside and inside is layer 6 deep. Now all incoming signals, they go to layer 4, and from layer 4 they spread up and down. Layer 1 and 2, they receive diffuse inputs from the lower brain centers. Layer 1 and 2 receive diffuse inputs from the lower brain centers. Now layer 2 and 3 neurons send axons to closely related portions of the cerebral cortex, presumably for communicating between similar areas. Layer 5 and 6, they send axons to more distant parts of the nervous system. Layer 5 to the brainstem and spinal cord, and layer 6 to the thalamus. And layer 6, 5 and 6, they are efferent. They go from there to the either brainstem and spinal cord layer 5, or more distant to the thalamus layer 6. Layer 2 and 3, they send impulses presumably for communication between similar areas. These are the layers. This is layer 1, outside, diffuse input, lower input. 2 and 3 are related brain areas. 5 are incoming, all incoming signals. 5 is to spinal cord and brainstem, and 6 is to the thalamus. These are the areas. These, of course, are histological layers. Cellular organization of the cortex. Within the layers of the neurons, there are the cerebral cortex are arranged into columns. So the cerebral cortex are arranged into columns. Each column serves a specific sensory modality. Say one column for stretch, one column for pressure, one column. So each column, like here, these columns here, and each column serves a specific sensory modality. Different columns interspersed among each other. They are interspersed so they can have certain communication between each other, and different cortical levels which allow the beginning of the analysis of the meaning of the sensory signals, because sensory signals might be interrelated. Now, destruction function of the somatosensory. Destruction of somatic area 1 results in loss of discrete localization ability. Loss of discrete localization is too bad. Inability to judge the degree of pressure. How much the pressure is. But still, if this is lost, still you might feel that there is sensation, but you lose its localization, you lose its judge the degree. Inability to determine weight of an object, or determine the what discrimination, and inability to determine the shape and form of an object called stereognosis. And this is bad for blinds, because the blind, they use this kind of sensation for reading. Unable to determine the shape and form of, it's called stereognosis. A for the nephi. Inability to judge texture, also the same like stereognosis, form and texture. Somatic association area. Behind each primary area, there are, say, in the lateral lobe, behind the lateral lobe, there are the secondary or association somatic area. The somatic association area has higher level of interpretation. It interprets the meaning of the primary sensation. Located behind the somatic sensory cortex and the parietal lobe. Association and receive inputs from the somatic sensory cortex, ventrobasal nuclei of the thalamus, VBL and VBM, and visual and auditory cortex. Now, function is to decipher the sensory meaning. What do we mean by decipher? Give a meaning for the kind of sensation. Give a meaning. What do we mean by a meaning for sensation? It makes a good localization, orientation from where is this area compared to the other area, and forming a three-dimensional position of the sensation. Three-dimensional position. Now, loss of this area results in inability to recognize complex objects. Three-dimensional loss of self, what we call it amorphosynthesis. So that person is unable to form a shape of the object it senses. ما إله قدرة إنه يكون شكل هذا الأوبجكت اللي it's going to be sensed to. So amorphosynthesis, an ability to form shape or recognize the complex object, shape of the objects. Why? Because the shape of the objects needs association of different kind of sensations, and this is lost because the association area functions in interpreting. It receives information from different parts and interpret it and give a meaning for the kind of sensation and form what we call three-dimensional ways. Special aspect to the sensory: thalamus has some ability to discriminate tactile sensation because some of the neurons they end in the thalamus, especially here for pain. Thalamus is an important role in perception of pain and temperature. So if there is damage to the post-central gyrus, still that person can feel pain and can feel temperature, but very little regarding tactile sensation, but still it can be. There are fibers that come from the cerebral cortex downward called corticofugal fibers. So these are descending fibers from the cortex downward, down to the spinal cord or down to the brainstem areas. What is the function of these fibers? This is negative feedback fibers. Fibers from the cortex back to the sensory relay, thalamus, medulla, and spinal cord. These fibers are inhibitory, and what they can suppress the sensory input. They can suppress it. They can remove this kind of adaptation. So this is kind of negative feedback, and they might go to the level of the sensory sensation to increase its sensitivity, increase the contrast, increase the sharpness, function to decrease the spread of the signal, to sharpen the degree of and contrast, to adjust the sensitivity of the sensory system. So these corticofugal, they go back from the cortex down, medulla or thalamus or spinal cord. They will decrease the diffusion of the impulse. So if they decrease the diffusion, they are important for sharpening of the impulse, or increase the contrast between on and off, and they can adjust the sensitivity. They will bring the sensory system down, the inhibited, to be at the level of the highest intensities and highest sensitivity system, highest sensitivity. This is some kind of coding. So if here this is the stimulus. If the stimulus is weak, then it is able to stimulate the central part of the cerebral cortical cells here. If this number of cortical cells are stimulated, then the impulse is perceived as weak stimulus. If the stimulus is stronger, so it's able to diverge and able to stimulate larger number of cortical cells. Larger number of cortical cells are stimulated. That person will interpret this as a higher intensity, and if it is very strong, it's going to stimulate even larger number of cortical cells. If larger number of cortical cells stimulated, it is going to be interpreted as very strong stimulus. So coding for intensity of the stimulus is perceived as a number of neurons that are stimulated, number of neurons. Besides the number of neurons, also is the number of impulses. We remember it that we took that coding for intensity. How the intensity is coded for by? It is coded by number of neurons you see here. The more the number of neurons of the cerebral cortex stimulated, the stronger the intensity and number or frequency of action potential. Number of action potentials or frequency of action potential. So this is the two ways by which the system codes for the intensity. Now the two bones discrimination, the two bones discrimination, it has a lateral inhibitory pathways. So this system inhibits laterally here, and this system inhibits laterally. So in the center here we have inhibition, and in the lateral we have here stimulation. So in the two bones discrimination is perceived as one stimulus here and one stimulus here, and in the middle it is inhibited by lateral inhibitory circuits. So processing of two bones discrimination involves at the cerebral cortex involves lateral inhibitory circuit. This inhibits lateral, and stays in the center, and this inhibits lateral, it stays in the center. So the central part of the two here is inhibited and the lateral parts are stimulated. You feel it as it is two beaks, one beak, in between there is inhibition and the other beaks. That's why how you code for the two bones discrimination. And graphical representation of the intensity, the stimulus strength. As the stimulus is stronger and stimulus, it is stimulated as it is going to be perceived as stronger and stronger due to the number of impulses. So here number of impulses. Note that the power law does not hold at either very weak, because here you got almost plateau, or very, this is the highest sensitivity. Any change in the strength here will have too much change in the number of impulses. So this is the highest in sensitivity area, highest sensitivity. This is low sensitivity, so high sensitivity here. This is here low sensitivity area. So here how you can discriminate position. Position is discriminated by a combination of, so look to here. If you are moving from this area to the extension, you are extending the limb. If you are extending the limb in the thalamus, there are neurons. When you move to higher degree extension, there are some neurons, say neuron 3, neuron 2, and neuron 1 in the thalamus. As you see, when you move this way, their number of impulses is decreasing, but the number of impulses at neurons 4 and 5 is increasing. So if you are moving this way and the number of impulses in lower in 1 and 2 and 3 are decreasing, and the number of 4 and 5 is increasing, the system knows that this is an extension. On the other hand, if it is here flexion here, the number of impulses at 1 and 2 and 3 is increasing, and the number of impulses at 4 and 5 is decreasing. Look to any degree of impulses. So degree 140 here. As you see at 140, you have this level of impulses at 4 at 5, and this level of impulses at 4. If you have this level of impulses, this number of impulses at 4 and this number of impulses at 5, your system knows that this is 140 degree of extension. If it say is 180 degrees, as you see 180 degrees has the same number at 1 and 2, but different number here at number . So here flexion, the blue here flexion مثلا, here can extension. So this combination will never have the same combination like each other. دائما هذا الcombination, this combination is specific for each degree. ما في combination, there is no other combination like each other. That's why this combination of impulses and these from this that comes from this neuron, this is example 5 neuron, but you might expect maybe 100 neurons around the joint, and these 100 neurons, some of the neurons are when you change the angle of the joint, some of the neurons they transmit more impulses, other neurons less impulses. When you move on the other hand, so the other neurons they increase their impulse rate, and the other neurons decrease their neurons. So this combination of number of impulses in these neurons, not just 5, maybe 100, 100 say 60 are increasing, 40 are decreasing, and so on, your system will interpret this as extension or flexion. Thank you very much. This is all what we have for this lecture.